Hello, my name is Dr. Roderick L. Roll, and today I will be talking to you about biology, the study of life. So biology is the study of life, and how we study life is we start from the smallest components of life and work our way up to the biggest components of life. In this slide, I have the hierarchy of life, starting from the biosphere, ending with the atoms. The first structural level of life that we will discuss is the biosphere. All life on Earth and the places that life will exist will make up the biosphere. So in this image, we have several places where life would exist. We have the tundra, we have the savanna, we have the wetlands, we have the coral reefs. So these are different places on the planet that contain life. So the biosphere is the places and the things. The second structural level of life is the ecosystem. This is a region within a biosphere where you would encounter life. One example is the wetlands. Fort Myers area in Florida resembles the wetland. So the wetland has the inanimate objects and life. So collectively all of that would be the ecosystem. So the third structural level is community. This is a group of different organisms in the Fort Myers area or the Everglades. We have the alligators, we have the egrets, those are the birds, and we have different types of mosquitoes. So all of those collectively are organisms that would reside in this area. They would make up a community. Now we will look at a population. A population is all individuals of a species. So all of the egrets collectively would be a population of egrets, versus number 5 is just one of those birds, and it is an egret. Now we will look at the organ systems, tissues, cells, the organelles within the cell, the molecules that make up the organelles, and the atoms. Within the human, we have 11 organ systems. They are denoted by the integumentary, skeletal, muscular, nervous, endocrine, cardiovascular, and so on. All of these are organ systems that help the human to function. One of those organ systems is the endocrine system. The endocrine system controls hormones. Some of the organs that would reside in this organ system would be the pituitary gland and the hypothalamus. They both secrete hormones. Growth hormones help cells to grow. So this is very instrumental in survival of the human. Now we will look at the tissue that makes up the organ systems. In the organ systems, you have several organs. Each organ is composed of layers of cells. These layers are called tissue. So one example of a tissue is skeletal muscle tissue. This tissue is involved in the skeletal muscle system. Number 8 in the structural level of life is the cell, the cellular level. One example of a type of cell is the nerve cell. You have cells that make up the liver. They're called hepatocytes. You have cells that make up the heart. They're cardiac cells. So you have many different types of cells in the human body. Within the cell, you have organelles. So a specialized structure that is within the cell is the organelle. Some examples will be the nucleus, the smooth endoplasmic reticulum, the rough ER, the ribosomes, and the Golgi apparatus. They all have their own individual functions. Each of those organelles are composed of molecules. Molecules will be a group of atoms covalently linked together or ionically linked together. Step 11, the smallest component of matter is the atom. So atoms make up molecules. Within the atom, you have subatomic particles, such as protons, neutrons, and electrons. To study biology, you have to understand what systems biology is. This is the approach where you learn the small, intricate details before you work up towards the bigger picture. So understanding the actual puzzle piece before you create the image in the puzzle. This is systems biology. So in this class, we're going to teach you starting at the chemistry level. We're going to start by teaching you what atoms are and how they interact with one another before we can move up to population of organisms and communities of organisms. Once we move to that level where we have many different types of organisms, you need to also learn what taxonomy is. This is how we classify all of those different organisms. So taxonomy is a filing system that is used to organize all the organisms on the planet. Biologists theorize that 10 to 100 million species of organisms exist, and we have only named and identified about 1.8 million. Now, out of these organisms, we have about 100,000 fungi, about 290,000 different types of plants, about 52,000 different types of vertebrates, and 1 million different types of insects. All organisms on the planet can be placed into groups based on similarities and relationship differences. Each group will include structural similarities and differences, functional similarities and differences, and other obvious features. For example, organisms that have 4 appendages can be all put into a group, whereas some of those organisms walk on 2 of the appendages where others walk on 4. So therefore you would diverge and make 2 more categories. Mr. Linnaeus came up with this classification scheme. This scheme put all organisms into 3 kingdoms with 6 smaller classification groups. The other classification groups were phylum, class, order, family, genus, and species. The way I was taught to remember this classification scheme is King Philip came over for good sex. Just make sure you remember what each of those structural levels mean. Kingdom, phylum, class, order, family, genus, and species. As technology progressed, grouping organisms changed. So in the past, all taxonomists had to use was visual characteristics like big, green, big, and moves. So if it was big and green and did not move, they place it in kingdom Plantae. If it was big and moved, they would say maybe it's an animal. Later on, the microscope was invented. So then classification had to be redone. Finally, we have new molecular approaches to classifying all the different organisms on the planet. Mr. Wolsey in 1977 came up with a new classification scheme. By utilizing rRNA, he was able to regroup different organisms that fell into the wrong categories previously. The next portion of the chapter we're going to discuss is the scientific method. It's also called scientific inquiry. Science is an approach to understand the natural world by asking testable questions. An addressable scientific question is, number one, testable, and number two, falsifiable. If it is not one of these two, it is not an addressable scientific question. Here is an example of nonsense. This is not a testable or a falsifiable hypothesis. Donald Trump is controlled by demons while on Twitter. This cannot be proven or falsified, so therefore it's nonsense. This answer is testable and falsifiable. Larry Wills is one of the strongest guys on the planet because he is on steroids. There's two experiments you can perform. You can analyze his urine or his blood for steroids. Number two, you can compare different people across the world and how much weight they lift. So this is testable and falsifiable. Scientists make observations and then form testable hypotheses. A hypothesis is a proposed explanation, a proposed answer to a question. The question is, why are there so many snakes on the campus of FGCU? The answer is, FGCU is infested with rodents. That is testable and it is falsifiable. So that is science. Justice for Harambe. This was a tragedy that occurred several years ago where a little kid fell into a primate enclosure. People freaked out, they lost their mind, and they killed the gorilla. The gorilla was protecting the kid. The kid was crying. The gorilla was protecting the kid. Now, after that is what upset most scientists. So at the heart of science is asking the question. This is called inquiry. Inquiry is search for information and explanation. So several months after that, we were still trying to address the question, was this monkey or gorilla supposed to die? Most of the individuals that got the audience, the largest audience, were the idiots who had no background in primate interaction with humans or primate interactions with other primates. The lady to the left is Jane Goodall. She's a primate expert. She wasn't seen on the news stations doing interviews. But to the right, these two dum-dums clearly had the loudest mouthpiece and the biggest audience. So therefore, their voice was heard. That is not good journalism when you just talk with no facts. So in science, we only talk with facts. Whenever you have a discussion with other students or even the professor, make sure you have your data, which will be your fact. If you have your fact, it's easier for us to digest what you're trying to say. So let's look at a simple experimentation. Observation-based inquiry with the flashlight experiment or example. This young lady is trying to use a flashlight and it is not working. She observes that it is not working. She's going to ask a question and then she's going to formulate two answers. The first answer is the batteries must be dead in this flashlight, therefore it's not working. The second answer is no, the bulb is burnt, therefore it is not working. So she will then run an experiment and test her hypothesis, test her answers. This is her testing her answers. The first result she gets, the prediction was wrong. The test falsified the hypothesis. Therefore, she moved to the second hypothesis. The bulb must be burnt. She fixed the bulb. She fixed the problem. So the test does not falsify the hypothesis in the second portion of the experiment. So the flashlight experiment was a very good example. Now let's see how this works in real life. You probably used this before in middle school or high school. You had to create a science fair project. There's 5 steps that we need to talk about. The first is you have to make the observations. You have to pay attention. You have to cut off the music. You have to put down the cell phone. You have to inquire. You have to observe. Once you observe nature, then you can ask relevant questions. So number 1, you have to make observations and inquire and ask questions. Number 2, you form logical hypotheses. These are logical answers to what you observed in nature. Number 3, then you would test your answer to what you saw. Step 4, you will collect data. And step 5, you will explain your data. So let's run through all of these steps. The first one is observing. Use your senses to make observations. Again, if you're always in your house playing PlayStation, you can't make observations of the nature, of the natural world around you. So you first have to free up some of the distractions and observe nature. Step 2, you will form a logical, tentative explanation for a question that you are trying to address. Step 3, you would design a procedure to test the answer to the question. This is called experimentation or methodology. Step 4, you will collect the data. There's different types of data. We'll discuss that. We have the social science data. This is very subjective. This is what you feel. In science, we don't care about what you feel. We care about what you see. This is the natural science. This is very objective data. So again, the psychologist or the social worker or the sociologist, they look at their data is subjective. It is unmeasurable. It is different to quantify, different to count. Versus natural sciences, we typically look at objective data. This is measurable. I can measure the amount of a neurotransmitter that is being released in the brain. That is something that is measurable. That is something that is precise. It can be quantified. So, 4A, objective, measurable data. This is data that can be seen, heard, or felt. I can feel that something is hot or cold. I can actually stick a thermometer under the tongue and take a reading. This is objective. This is precise. This is accurate. I could take the blood pressure. This talks about how much pressure is being exerted on arteries and veins. So examples of objective data is taking the temperature or vomit. If someone vomits on you, that is a positive indicator that they might be sick. Versus if someone just tells you that they're sick, that's subjective. I don't care about feelings. I care about data, objective driven data. So here is subjective data. That is unmeasurable. You hear terms like pain, feelings. That is subjective. This is information perceived only by the affected individual. This is what has created our opiate epidemic in the United States. We have a crisis going on because doctors are overprescribing this medication for pain. Why? Because if you ask a patient what is their pain level, and if they say a level 10, they will receive the drug, and the drug makes them feel very good. They're in a euphoric effect. Then therefore they will lie and say that their pain is level 10 so they can receive the opiate. So that is why we're in this crisis now. We pay attention to too much subjective data and not enough objective data. 4C is qualitative data. Qualitative data is unmeasurable. It's recorded description using words such as soft, firm, hot, or cold. We have to also be mindful and be careful of using these terms too. When a patient comes in and you say they are hot or they are cold, that doesn't really tell the doctor anything. We need numbers. If the number is 98.6 and you say they're hot, what does that mean? 98.7? Or does that mean 104? So again, you have to be mindful of the type of data that you give the doctors and so on, and scientists. 4D is quantitative data. This is measurable. This data will typically be drawn in graphs and charts. This is recorded description using numbers, typically seen in tables and graphs. So after you have collected your data, we know the different types of data, then you would have to explain your data. This is typically referred to as the discussion portion or the conclusion. We can also look at data and separate it into inductive reasoning and deductive reasoning. We'll first look at inductive reasoning. This is a set of observations that helps you reach a conclusion. This is an example of inductive reasoning. For centuries, people looked at cells under microscopes in different organisms. After viewing many different organisms, biologists came up with the cell theory that states all organisms are composed of cells. How did they come up with this theory? Through observations, through many decades and centuries of looking at tissue from different organisms and seeing the same thing. So the cell theory states that all organisms are made of cells, from this smallest mammal to the right to the largest mammal to the left. all organisms are composed of cells. And we know that the cell is the smallest unit that contains life. Anything smaller than the cell is not living. Organelles are not living. Viruses are smaller than cells. They are not living. So this concept was developed in the 1800s, and it is also called the cell doctrine, that all organisms are composed of cells. This doctrine was an inductive generalization based on microscopic examinations of plants and animals for decades to centuries. Who first saw these cells? Robert Hooke. So in 1665, Robert Hooke discovered the cell, and he published this. He described the dead cell wall of plant cells as it appeared under the microscope. Mr. Leeuwenhoek, later on in 1674, described a living algae called Spirogyra. We will look at Spirogyra in lab. Mr. Leeuwenhoek also was the first person to see bacteria. Why? Because he was able to take glass and grind it down in such a fashion to make these very, very powerful microscopes. So before 1674, people were not able to make that great of microscopes to be able to see bacteria, but he was. Mr. Swann and Mr. Leiden. In 1839, Swann wrote, "All living things are composed of cells and the products of the cells in their structure." Both of these two individuals came up with this theory in 1838. Mr. Virchow wrote a book, and in his book he said all things arise from pre-existing cells. This doctrine negated the theory of spontaneous generation, like previous scientists like Aristotle stated. They thought that organisms spontaneously appeared. If someone was living in sin, they could spontaneously get syphilis. We now know that is not true. Why? Because Virchow theorized that all things arise from pre-existing things. He performed an experiment that proved that. Now we will look at deductive reasoning. This is if-then logic. Here is an example of deductive reasoning. If all organisms are made of cells, and humans are organisms, then humans must be made of cells. Let's look at how an simple experiment is laid out. You observe something, you come up with an explanation for it, you design an experiment to test your explanation. You conduct the experiment, you look at the data, you collect the results, and then you draw a conclusion. If this matches what you expected, then you perform it over and over again. You have to make sure the data is tight. You have to make sure the data is consistent. So that is how to perform a simple experiment. We're now going to look at an example of how a dum-dum did not perform that simple elementary experiment correctly. So here's a question for you. Does the MMR vaccine cause autism? What is the MMR vaccine? Measles, mumps, rubella. It's a vaccine that you receive early on in life. First shot is between 12 and 15 months. The second shot is between 4 and 6 years. And the third shot is 18. Most of y'all Probably just took your shot. Majority of the scientific community will say no, MMR vaccines will not cause autism. But a dum-dum several years ago falsified data to fit a narrative to get published and to get funding, grant funding. We're going to talk about his experiment. So let's look at the answer that most people received. MMR vaccines do not cause autism in children. Majority of the scientific community, no, let me say this, all of the scientific community received the same answer. Therefore, no one performed this experiment. Then comes another scientist. He had a different answer. MMR vaccines cause autism in children. So, he performed an experiment that proved everybody else was wrong. That experiment subsequently ruined his career. So in 1998, Mr. Wakefield published a fraudulent research paper. And a lot of people jumped on the bandwagon. You know, remember those people with the loudest mouth and the most followers. A lot of people jumped on the bandwagon and believed it. They thought that measles, mumps, and rubella vaccine causes autism. He was subsequently proven wrong. Papers were retracted, lost his grants, and he's not working as a scientist anymore. So the next couple of slides, I want you to read through an experiment that was in your book. It talks about predation of snakes. So here was the question: does the presence of venomous coral snakes affect predation rate on their mimics? So a coral snake has a very, very deadly bite. The coral snake is when red touch yellow. When the red stripe touches the yellow stripe. When the black stripe touches the red stripe, that one is not poisonous. So the question was, the king snake is going to be protected because it appears, it mimics the coral snake. So that was their question, that was their answer, and they devised an experiment to test that. You need to read through so you can see a real life example of science at work. This slide shows the artificial king snake that was fake versus the artificial brown snake that was fake. What they looked at to see if raccoons and bears and cougars would destroy either of the snakes. And they saw that the artificial brown snake got mutilated and destroyed when it was seen. So read through the experiment on your own. These are the results from that experiment. This is the rest of the results from the experiment. So we're going to be looking at a lot of experiments, so we need to understand how to perform a controlled experiment. So a controlled experiment is one that is designed to compare an experimental group with a control group. That is a controlled experiment. A control group typically does not receive the treatment, versus the experimental group will receive the treatment. So in drug studies, one group will get the placebo, the fake drug, the one that has just sugar or water, whereas the other group will get the experiment, the actual drug itself. These are two other terms that we will get familiarized with, especially when we get to the macromolecule chapters. We will need to know what a positive control is. This is the sample that will give us our expected response, versus no response is expected in the negative control. Typically, when we perform our experiment, water will be used for our negative control. A few more terms we will learn is independent variable versus a dependent variable. The independent variable are manipulated by us. We are the researchers. So in this example to the right, adding water is the independent variable. So liquid used in growing a plant would be the independent variable. If every table had a plant and table 1 added 10 milliliters of water to the cannabis plant, table 2 added 20 milliliters to the cannabis plant, table 3 added double that, 40 milliliters, we could measure leaf growth in these plants based on the amount of water we supplied. So the water being supplied to the cannabis plant would be our independent variable. That is the thing that we change. The dependent variable is what we measure, how big the leaf is, or how strong or how much THC is produced. So that is a independent variable versus a dependent variable.